All right. Last time I talked about the concept of homeostasis, which is what? What is homeostasis? Somebody give me a rough definition. Think back. Oh, a long weekend. It was a long weekend. What did I say on Friday? What is homeostasis? Anybody? You can read your notes. I don't mind. Go back. Yeah. Yeah, right. So the definition I gave you of homeostasis was a mechanism that seeks to maintain a stable internal environment, right? The loss of which can result in illness or death. Okay. And I started talking about negative feedback, and I gave a few examples of that. One of them was the postural change of blood pressure, right? You stand up and blood drains from your head because you've been asleep all night, and the baroreceptors in the carotid bulb and the aortic arch sense that rapid drop in blood pressure in the upper extremity and send a signal to the brain, the cardioacceleratory, not the brain actually, the brainstem, and then the cardioacceleratory region of the brainstem sends a signal via the vagus nerve to the heart, speeds up the heart, and raises your blood pressure. All right, and if this doesn't happen, then you can black out. Right, and I talked about how this happens to older folks. That was an example of this that I gave. Another example, the first example I gave was temperature regulation in the body, trying to keep the body at a stable internal temperature. Right. So, for example, I used to work at a camp for children with chronic skin disease, and they're sponsored by the American Academy of Dermatology. And there was a condition, an ichthyotic condition that some of the children had that had an accompanying symptom, anhidrosis, meaning that they couldn't sweat. These children couldn't sweat, which is a bigger problem than you may imagine. Has anyone had a dog before? Right? You don't leave your dog in the car, because that dog can't really sweat in the same way we do. They blow most of their heat off through their mouth, which is why they do that. Well, these people had a similar problem, and if they were as normal kids that they are, running around, they heat up, their body generates heat, but the mechanism for removing that heat is not in place, and it can be quite dangerous and deadly for some of those people, right, to not pay attention to that. So this is homeostasis, right? Maintaining a stable internal environment. Now, today we're going to talk about, and that was negative feedback, where the influence of the effector opposes the change in the variable that we're seeking to control, right? Maybe temperature is going up in the body, and the effector is going to oppose that by sweating or what have you, or maybe we're cold, the temperature is going down, and so the effector is the muscles is shaking a vibration that is going to generate heat in the body, for example. Positive feedback homeostasis is still trying to maintain the health of an individual, but a positive feedback system is something in which there is a process that's happening, that needs to happen, that is self-amplifying. All right, it's something that when it begins to happen, it causes itself to happen more. It's like a cascading effect. All right, sort of like trending, something trending on social media or whatever. It sort of exponentially explodes. So here's some examples of positive feedback. And I guess before examples, the definition I have here is that the response of the effector, contrary to negative feedback, the response of the effector increases the change in the stimulus. So the body is moving away from some sort of normal, stable situation. All right, hopefully to return at some point. Whatever normal range in the variable that's getting controlled is going to be lost. This is a healthy way to produce rapid changes that need to happen sometimes. So here's several examples that'll help you understand this. Hemostasis, don't be confused, that's not homeostasis. Hemostasis is the word that describes the stopping of bleeding. When your body is bleeding and you need to stop that, it's called hemostasis. So what happens? We have a break in a blood vessel. We get a cut of some sort, a rupture of capillaries or arterioles, hopefully not an artery, but something breaks that's carrying blood. And the cells that were damaged in that cut release all kinds of chemical factors that initiate clotting. The clotting cascade is sometime when you're bored but curious, you can go to Google and look at the clotting pathway. I'm certainly not going to touch it here today because we could probably do a whole course on clotting. But it's probably one of the most complicated molecular pathways you're likely to run into in medical school if you go there or whatever. But suffice it to say, we're going to treat it as a little pink box right there. The clotting begins. And there are several factors that get produced in this clotting cascade that speed up clotting. They're self-amplifying factors. We're going to see a number of these throughout the semester. Because when you get a cut and you trip the trigger, the body immediately recognizes, you want the body to immediately recognize that there is a problem and you want to stop fluid loss as quickly as possible. So you want to have an exponential growth in the ability of those clotting factors to get engaged on site in the place of the injury. So eventually, you're going to form a blood clot. We're going to activate platelets. We're going to activate fibrocytes. Fibrinogen's going to get hydrolyzed to fibrin, and we're going to form a clot, a cell patch, a cellular patch that will stop the bleeding. And when that happens, we're going to slowly ramp down, right? The clotting factors that started this positive feedback loop are eventually going to be turned off as the damage is sequestered. All right. There's a lot of examples. Contractions during childbirth are one. Protein digestion. When we get to the GI chapter, I'll talk about trypsinogen. Trypsinogen is a proenzyme, an inactive enzyme that has a little regulatory tail on the protein, on the peptide chain that will get cleaved off. And when that happens, trypsin is going to be able to break down other proteins into the amino acids, but by cleaving peptide bonds. One of the great things about trypsin is, however, it converts trypsinogen to trypsin. It's kind of like if you've ever seen The Matrix, that movie with Keanu Reeves or whatever, the spooky secret agent guy or whatever, right? That like comes up and just touches somebody else and turns them into him. It's kind of like that. Trypsin is able to catalyze the conversion of trypsinogen into more trypsin. All right, so that's a positive feedback mechanism. Fever is another one. So as when fever gets triggered and your fever begins to go up, the fact that your fever is going up can accelerate the process of raising your fever. All right, so that's positive feedback. Ovulation, the release of an egg. Lactation, when the mother has milk let down, the fact that she is letting milk down from the mammary gland stimulates the release of more milk for the baby. That makes sense. Action potentials, the generation of nerve signals. These are all positive feedback mechanisms. So here's contraction. The baby, it's time to go, and the woman starts having what are called Braxton Hicks contractions. Has anyone been around when did I ask you guys this already? Yeah, babies. If maybe you were around your mother or your aunt or your older sister or something when she was having a baby, you know that in the day or so leading up to the birth, they begin these irregular, mild contractions called Braxton Hicks. And that kind of wakes the uterus up to the fact that it's going to be time to deliver the baby. The baby descends into the it drops down into the pelvic bowl, and the head of the baby presses upon the cervix, right? That pressure on the cervix sends signals up to the brain, afferent signals up to the brain, which say, ooh, looks like it's time to have a new one in the world. And the pituitary gland produces oxytocin, which enters the bloodstream and has a number of effects. Oxytocin is a very powerful hormone that has a lot of effects. But one of them is to stimulate uterine contractions. And the uterus contracts in the presence of this wave of oxytocin that has been released because of the pressure from the previous contraction. And it causes a more intense contraction. And the head pushes even more firmly on the cervix. And with increased pressure, you have a stronger afferent signal to the brain. And with a stronger afferent signal, we get a stronger release of oxytocin, and so on and so forth. You can see how that can build until finally the baby descends into the birth canal and is born. All right, beautiful thing. Okay. There are others. There are many others. Example to make men not feel like they're left out. Ejaculation is an example of a positive feedback mechanism. All right. So for any of this to happen, there has to be communication, right? I talked about there being intrinsic and extrinsic regulation, didn't I? Intrinsic regulation is one, or autoregulation is one in which the tissue itself is regulating itself, right? But extrinsic regulation is essentially dependent upon one of these two forms of communication, either the nervous system or the endocrine system. Nervous system being electrical, where you have a neuron that is communicating most likely with the central nervous system somewhere. And then on the other end, the downstream end, with some sort of effector cell, right? Whatever that is, whatever the effector is. And then a chemical communication is via the endocrine system. So the endocrine cells are going to produce some kind of hormone that gets released into the blood, circulates, and then is going to hit the target cells. And those target cells can be widely dispersed. So these two forms of communication have some implications, though, that we'll see here in just a moment. So whether it's, I guess I began talking this semester about structure-function paradigm, right? And whatever function a system is meant to serve is going to be dictating the structure that is there. All right. So the requirements of that function are threefold: distance, speed, and distribution. So first of all, distance. How far away from the effector that the cells that are causing the change that we need, how far away from the effector is the integration site, the integration center, all right? Is it short? If it's short, then it can be, it may be an endocrine form of control, where we have either autocrine, which is that intrinsic regulation that I talked about. Autocrine are cells that are regulating themselves, or paracrine are just cells that are next door neighbors. They may be in direct contact with one another, in fact. But closely aligned cells, paracrine or autocrine. So this is for short distance. Long distance, maybe it's better to use somatic nerves of the body, the peripheral nervous system. Maybe the integration center somewhere in the brainstem, and you are trying to control the adrenal glands or something like that, or the suprarenal glands, and that's going to be a nerve, a longer nerve. The speed of communication. Is it a process that has to happen rapidly as soon as we sense a problem, or is it a longer or slower process that's happening? If it's fast, then perhaps the nervous system is the right way to go, because it's very rapid. It's a very rapid form of communication, whereas the endocrine system needs to rely on the cardiovascular system to distribute those chemicals throughout the body. And then finally, distribution. Is the effector that we're concerned with in whatever homeostatic system, is it a single muscle cell or a small group of cells that can be in direct contact with a nerve, right? If you want a very focused form of control, the nervous system is the way to go. Whereas if it's more diffuse, if they're, like, for example, in the childbirth example that I talked about, there was the effect of oxytocin on the uterus. But oxytocin does a lot of other things, right? It reduces the woman's experience of pain, I guess is the best way to describe that. I've never experienced it, of course, but it has a number of different functions, and it's going to attack different, attack is not the right word, but it's going to cause an effect, elicit an effect in a variety of tissues in the body. So the endocrine system, a chemical form of communication, is probably the best way, because the blood perfuses the whole body, and if the hormone is released into the bloodstream, it's going to be widely perfused. Okay? All right. So that's where I had wanted to get last time. This is the beginning of what I had intended to talk about today. So now I'm going to talk about one specific form of homeostasis in more detail, specifically how the body regulates calcium. I'll have to do that. I'm going to have to tell you a little bit of anatomy. This isn't an anatomy class, but there's going to be some anatomy that we need to learn so we can talk about it. I'll go through the osteocytology, basically the bone cells, the cells that make up a bone. I'll talk a little bit about the matrix and marrow, not much. But then I'll get into how we discuss the elements of calcium homeostasis. So calcium homeostasis is this sort of rule of threes, right? There are going to be three hormones that dictate it: calcitonin, parathyroid hormone, and calcitriol, in three tissues: the bones, GI tract, and kidneys. I'm not sure if I'm going to get to the pathology today, just because I had to make up some time. All right, let's dive in. Calcium. So I'm sure everyone here is at least vaguely aware that calcium is an important nutrient in the body, right? And you're probably also quite aware that calcium helps build strong bones and teeth, because that's probably been drilled into your head by your parents since you were a little boy or girl, telling you to finish your milk or whatever it is, so you can get the calcium you need. Calcium does a lot besides just build strong bones and teeth. It of course does that. But calcium is extremely important as the trigger for the molecular trigger for a process called exocytosis. What is exocytosis? There's a little picture there that'll help stimulate your recollection. Have you talked about exocytosis? Did they talk about that in 163 or 4 at all? What is it? Come on, give it a guess. Take the word apart. What I'm trying to do is get you to take the word apart. Exo, outside, cytosis. Sending something outside of the cell. All right. Exocytosis is the process of which cellular contents, some sort of thing that the cell made, some cellular product that has been packaged in a vesicle, a membrane-delimited vessel. You guys have talked about plasma membranes before, right? Is anybody not familiar with the concept of plasma membrane? Don't be shy. I don't want to. Okay. A membrane-delimited vesicle that's full of cellular contents of some sort. That vesicle is going to have the membrane fused with the outer membrane of the cell. And in so doing, what is on the inside of the vesicle, but excluded from the cytoplasm, now becomes released into the outside world. All right? You see that? A little process I have showing there. And you can see an electron micrograph on the right-hand corner up there of this actually happening. So exocytosis is the process of releasing cellular contents. There's a and there's many examples of this, right? Like all glands do this. Like many cells, immune cells do this. An important example of this is neurotransmission. So neurotransmission involves the release, the exocytosis of secretory vesicles that contain neurotransmitters, whatever that neurotransmitter may be. There's lots of them, into the synaptic cleft. So that's a special example of exocytosis, which is triggered by calcium. I talked about how complicated the clotting cascade is. Calcium is an important trigger in the clotting cascade as well. When we talk about muscles, we'll see how important calcium is to muscle contraction. It's essentially the trigger inside the muscle cells that allow muscle fibers to contract. Extremely important to like all fundamental processes in the body. It's hard to find tissue that doesn't do something that isn't exquisitely dependent upon calcium. All right, so because of that, it's extremely important to be able to regulate the calcium levels in your body. Let's talk about how much is there first. So in a typical human, a typical adult body, it's about a kilogram, a little over a kilogram of calcium in the body, 2.5 pounds. And the vast majority of that, 99%, is in the skeleton. Just because it's in the skeleton doesn't mean that it's stuck there and it never turns over. In fact, 18% of the mass of your adult skeleton is exchanged with the blood every year. Every year. Your body is constantly breaking bone down, building bone back up. So almost a fifth of the mass of your skeleton is eroded and rebuilt every year. Your plasma concentration of calcium is about 10 milligrams per deciliter. A deciliter is kind of a large shot glass, maybe a little juice container, a little juice glass, a small juice glass would be a deciliter. So about 10 milligrams of calcium in that volume of blood. And down in the bottom, I have this table. You don't need to memorize all these numbers. Maybe I should get rid of the bulk of those. But I put them there just to show what's in the green box on the end, that half of the 1% of the calcium that's not in your skeleton, this 10 milligrams per deciliter, about half of that is just free calcium floating around, free calcium ion in the blood. But about 44% of it is bound to some sort of protein. Some kind of protein is folding around it or has somehow bound it to transfer it. And a small amount of it is actually complexed or chelated. All right, so functions of the skeleton. Do I want to spend that much time on this? Yes, sure, why not? Support. You guys knew that, right? What else? What else does the skeleton do? I stole the easy one from you. What else does the skeleton do? Yeah, say it. Yeah, movement. Good. Oh, well, yes, movement and protection. Nora, I try to put these in the order that I suspect people to guess them in, but sometimes I'm wrong. So yeah, movement. There you go. It was number 3. It's like that show. What was that one where they like guess what's up there? I don't know. What was it called?
Top 5.
Yeah. Family Feud. That's right. It's like Family Feud. Electrolyte balance is the next one. So I've already said that 99% of the calcium is stored in the skeleton, but almost 20% of it turns over every year. So it's going to be involved in electrolyte balance, different charge carriers in the blood. All right. Acid-base balance as well. It kind of goes along with electrolyte balance. The skeleton helps to maintain the proper pH. And the red bone marrow is the site of erythropoiesis. Erythropoiesis is the process, I've used that word before, I think, in here, is simply the process of making new red blood cells. All right, so that's what the skeleton does. Now, we're going to have to learn a little bit of anatomy here. Bone itself, we're going to take a cross-section of a long bone. There's 6 different types of bones, and one of them being the long bones. So here's a cross-section of a long bone, and there is, on the outside of the bone, there is this layer called the periosteum, which has 2 layers, 2 different cellular layers. A fibrous layer and a osteogenic layer. That just means a layer where new bone cell formation happens. Then there's a similar layer on the inside of the bone, on the marrow cavity, and we call this endosteum. Periosteum means along the outside or surrounding the osteum, referring to bone. And endosteum just means covering the inside surface of a bone. And there's this layer of reticular connective tissue. We haven't talked about the different types of connective tissue yet, but we will. We'll get there. These are the 2 cellular layers where the cast of characters that I'm going to introduce to you live. So let's just look at this little zoom in here, this little magnification of the endosteum. We see the dense bone, this dense bony matrix right here. And surrounded by this dense bony matrix are osteocytes. These are the mature bone cells. Then on the actual endosteal layer, we have 3 different types of cells. Osteoprogenitor cells. These are a partially differentiated cell, kind of like a stem cell. It's like a stem cell just for bones. All right. We'll talk about exactly what that means in a moment. But osteoprogenitor cells can become osteoblasts, which are also not terminally differentiated cells. These osteoblasts are much more numerous. And then there's a third cell type on this endosteal layer called an osteoclast. Clast. Osteoclasts are not related to the other 3 cells that I've pointed out. Okay, and we'll talk about why in a bit. All right. So first, let's go through the 3 that are related to one another. Starting here at number 1 in the middle is the osteoprogenitor cell. Osteoprogenitor cells are derived from embryonic mesenchyme. I didn't go through the embryology tissues here just to save myself some time, but it's comes from embryonic mesenchymal cells, and they get stimulated. They get called into action when there is some sort of stress on the bone. Maybe there's been a fracture, maybe you have begun to do some weight-bearing exercise, and you're putting pressure on the bone. When this happens, so it's actually a pretty interesting process. Bones themselves are slightly piezoelectric, so the actual force compressing the bone creates a piezoelectric effect that stimulates these osteoprogenitor cells. When they get activated, osteoprogenitor cells, their only job, the thing that's distinctive about them is they can divide. They can divide into new cells. They split and divide. Mitosis. They go through mitosis. And when this happens, these divisions that they calve off, those divisions mature into what are called osteoblasts. When you're thinking about osteoblasts, you're going to be comparing them to osteoclasts. Osteoblasts are non-mitotic. They cannot divide, but the B in blasts, in my mind, says build. They build bone. Osteoblasts build bone. Lots of B's to stick in your brains. Osteoblasts build bones. In fact, what they do is secrete this organic matrix, the matrix, that it's going to be the job of the osteocytes to ossify or calcify and turn into actual dense bone material. All right, but the osteoblasts build the osteoid, which is this matrix that is going to get calcified and turned into bone. They produce a hormone called Osteocalcin, and it's an endocrine hormone that has a number of functions. It's going to increase the production of insulin by the pancreas, and it's going to increase insulin sensitivity in adipose tissue. All right? It's going to increase insulin sensitivity in adipose tissue, which is one of the reasons why exercise, one of the many reasons why exercise is one way to help prevent insulin insensitivity, which is the precursor to diabetes. All right. So osteoblasts can then develop into osteocytes, these mature bone cells, which are also non-mitotic. They can't divide. The number of osteocytes is purely dependent upon the number of osteoprogenitor cells you have. And the job of the osteocyte is to calcify or deposit a bony matrix. And they help regulate calcium and phosphate homeostasis. They live in these structures called lacunae. The word lacunae, L-A-C-U-N-A-E, plural of lacuna, without the E on the end. It just is a general anatomical term that just means a cavity or a hole, a space, a void space. All right. Cavity is not the right word because a cavity has an external opening. A lacuna is a fully encapsulated hole in some sort of dense surrounding. All right. Then on the other side, we have osteoclasts. And osteoclasts are these cells, it's like the blob that ate Pittsburgh or something like that. They're these huge cells that have as many as 100 or so nuclei, a polynucleate cell, and the job of the osteoclast with a C is to corrode the bone. All right, it dissolves dense bone. Okay, osteoclast corrodes bone. Class C, corrode. Hopefully that helps you remember that. All right. So they're formed from a number of cells, these megakaryocyte cells, that stem cells that fuse together and form this multinucleate blob with this brush border, this ruffled end that embeds itself into the endosteal layer and is able to dissolve the bone. So when we think about that 18% of bone that's turned over every year, it is the combined and balanced activity of osteoblasts building bone and osteoclasts dissolving bone. All right, does that make sense? So whatever remodeling happens, say for example, you decide to take up bodybuilding. You decide to take up bodybuilding, and you are whatever activity, maybe let me think of some kind of bodybuilding activity. Yeah, so you're going to start doing curls, right? You're going to do curl, and you've assiduously avoided this your whole life, right? It's like, oh, I just want my arms to rest. That's all they want. And then you take up bodybuilding, right? You start doing this. If you were to take an X-ray of your elbow, the antecubital fossa, that included the radius, the head of the radius, you would see that before you started bodybuilding, the radial tuberosity would be pretty small. That the muscle that pulls the bicep and brachialis muscles that pull on the bicipital tendon and the radial tuberosity hadn't really done much to the bone. But after a year of just building, what was that movie? A Benicio del Toro's movie, not Shape of Water, one that I had a long time ago, but I think it was about a mermaid that lived in a pool. Did you guys ever see that movie? There's a funny character in there that hangs out in the stairwell of this apartment building, and he just does curls in one arm, not the other. He's just really into curling in one arm. And so this character has this enormous arm on one side and this like scrawny little nothing on the other. So if you were to compare that person's skeleton, the skeleton that was huge would have a big radial tuberosity, right? And the growth of the bone there is dependent upon the action of the osteoblasts and osteoclasts. So if that person were to have built a larger radial tuberosity on the arm that was working out, which of the two bone cells, osteoblasts or osteoclasts, would have had to have been a little more active in that year? Osteoblasts, because it built a little bit surplus bone. Make sense? All right. Bony matrix. We're going to skip this. This is important. I think I'm going to skip this. Yeah, I don't think I need this. Oh, I do. Yeah, I do. Sorry. So the bone itself is made, about a third of it, the dry matter, is made of organic matter. All right. So this is the matrix, the osteoid, that's laid down by which cell type? Which cell type makes the osteoid? Which cell type builds the osteoid matrix for bone? Build osteoblasts. All right, so this matrix is getting made by the osteoid's getting made. This organic matter is getting made by the osteoblasts. I guess I say it right there, don't I? It says osteoblasts. Well, what is it? It's mostly collagen, which is a protein, strong sort of Hauser rope of the protein world. And then this large family of carbohydrate protein complexes, the glycosaminoglycans, proteoglycans, glycoproteins. I would love to dive deeply into that, but the carbohydrate chemist in me shudders and moves on. Then there's the inorganic matter. 2/3 of the dry weight of a bone is made by the osteocytes. And this is essentially a ceramic. It's a ceramic. And we see right here, this is a model of what this is. 85% of this ceramic is called hydroxyapatite. Hydroxyapatite. It has quite a bit of calcium, but you'll notice in there that to make hydroxyapatite, what other elements do we need? What other elements, what other ions are in there besides calcium? Well, quite a bit of phosphate, isn't there? There's quite a bit of phosphate. The phosphate is actually why I decided to not skip this slide. Hydroxyapatite requires both calcium and phosphate. And then about 10% of it is calcium carbonate. This calcium carbonate is going to be the component in the bone that helps regulate the pH, because carbonate is going to act as an alkaline buffer in the blood if you're going to liberate calcium carbonate. All right. Guys, follow all that. I hit the important parts there. There's a lot of information on that slide. I guess I did the minor point that, it's not I guess that minor, but there are a lot of other trace elements. There's magnesium, sodium, potassium. Big boy is phosphate next to the calcium. All right. So marrow, yeah, I actually probably could have skipped this slide. I debated keeping it. In children, marrow is, the red marrow is found throughout the body in both the long bones and in what's called the axial and the appendicular skeleton. So the axial skeleton, there's a little bit of anatomy for you. The axial skeleton is the head, the vertebral column, the hips, and the rib cage. And it's axial because it goes down the axis of the body. The appendicular skeleton are your appendages. The appendicular skeleton, your arms and your legs. In children, all of those bones, yeah, come on, here's one. Here's an example. Right on cue. Thank you. Send him in. So in this specimen that you see right here, this guy who has the flu, you can just sit in the desk wherever you want. In his body, all of his bones, his long bones, his ribs, everything has red marrow in it. This is the tissue that makes red blood cells. It has goes through erythropoiesis. However, in my body, Exhibit B, the long bones, the marrow in the long bones has not turned off. It's just sort of changed. It's not red any longer. It's not erythropoietic tissue any longer. It's actually just fat. It's turned to yellow fat. Gotten old and fat in my bones. Yeah, it doesn't produce blood any longer, although the axial skeleton continues to. And it's at this time of life, right here, when you stop adding significant body tissue, when you're not growing any longer and blood volume is not increasing, that conversion happens. All right, so you guys at your stage of life are a little bit more like me than like him. Maybe sadly or happily, I don't know. Okay, so here's the 3 and 3 that I talked about earlier. Calcium homeostasis is going to be regulated by 3 hormones and 3 tissues. The 3 hormones are calcitonin and parathyroid hormone and calcitriol. Calcitonin is made by the thyroid. Parathyroid hormone is made by the parathyroid glands. So the parathyroid glands are these little lentil-shaped glands that you see on the back of that butterfly-shaped gland up there. The thyroid gland is just above your thyroid cartilage. It's right here in your neck. And on the back side of the thyroid gland are those little yellow spots called the parathyroid glands. This is where parathyroid hormone comes. Calcitriol, vitamin D3, comes from a number of places. It originates as vitamin D3 in the skin, and then goes to the liver and the kidneys for eventual conversion into calcitriol. So that's where those three come from. And then calcium itself is going to be regulated in three essential places. First of all, in the bones. This is where 99% of the calcium is, right? 99% of the calcium in the body is stored in the bones. So this is the site of storage. The second important place is the digestive tract, your intestines. This is where calcium is going to be absorbed, and we can these hormones I'm talking about are going to affect the ability of our GI tract to absorb more or less calcium. And then lastly, the kidneys. So GI tract is how calcium gets into the body, and the kidneys are how it gets out of the body. And affecting the way the kidneys work, we're going to either excrete more calcium into our urine or less. All right, does that make sense? So that 18% of the skeleton that's turned over every year and is replaced by calcium that you consume, some of the calcium that's in your urine came from your bones. You are literally urinating your bones away. It's kind of amazing. All right. We have just a few minutes more, and I'm going to begin by talking about the specifics of how these hormones work. So let's start with calcitonin. So calcitonin, the way you remember this is perhaps your serum level, the blood level of calcium is too high. Let's start in that case. We've deviated from homeostasis, and the calcium, there's too much calcium in your blood. It's too high. So we're going to tone it down. We want to tone it down with calcitonin. All right? Help you remember that, maybe. What does calcitonin do? It gets secreted, and it's going to have two effects on the bones. First of all, it's going to turn off, and you could probably guess this. You could probably guess this, knowing that it's trying to lower calcium in the blood. It's going to turn off osteoclasts, right? Because what was the job of the osteoclast? To corrode the bone, dissolve the bone, and liberate calcium, right? So it's turning them down. And it's going to turn osteoblasts up, right? So all this calcium that's in our blood is going to get stored. There's going to be a net storage of calcium in the bones. Calcitonin also allows you to urinate more calcium out. It allows for more excretion of calcium into the urine. It's really PTH that has an effect on the intestines. We'll talk about that in just a moment. So calcitonin comes out of what are called the clear cells of the thyroid. And so it's the thyroid that is responsive to ambient calcium levels. All right, parathyroid hormone. I can probably get through this slide. So now, parathyroid hormone and calcitriol are the other side of the coin. It's the other side of the coin. So now maybe our blood calcium level is too low. We do not have enough calcium in our blood. And there's going to be a host of things that parathyroid hormone elicits. So first of all, it's going to be the exact opposite of the cellular effects on the bone, right, on the storage system. It's going to turn osteoclasts up. It's going to turn osteobuilding cells down, the osteoblasts down. As osteoclasts get turned up, we're going to dissolve bone away and release more calcium, and osteoblasts get turned down. We're going to build less bone, taking up less of the calcium that's in the blood. Importantly, less calcium is going to get excreted into the urine under the effects of parathyroid hormone. So we're not going to urinate out as much of the calcium. But now this one is a little bit confusing for sometimes. More phosphate is going to get excreted into the kidneys, though. So we're ridding ourselves of more calcium, right? No, I'm sorry. We're preventing ourselves from ridding ourselves of more calcium. We're retaining more calcium from the urine, but we're ridding ourselves of more phosphate. We used phosphate to build bone. Why are we getting rid of it? What do you think? Who's had general chemistry here before? All right, most of you. Those of you who had, do you remember the word stoichiometry? Does that suggest a way forward here? If we're getting rid of phosphate, we need phosphate to build bone, right? We need the phosphate to build the bone. And if we don't have the phosphate in the correct stoichiometry for the calcium, then we cannot store calcium in bones, all right? So by getting rid of phosphate, we're able to leave some of the calcium on the table, so to speak, in the blood, all right, and elevate our blood calcium levels. So we are retaining calcium in the urine, but losing phosphate. We're trying to throw off the stoichiometry of phosphate and calcium in the blood. Does that make sense? Does that make sense to anybody? No? Yes? Everybody? All right, well, that's it. We'll pick up next time with calcitriol, vitamin D3. And maybe one of these days I'm going to catch up that 10, 15 minutes that I'm behind in my PowerPoints here.